Hi everyone, my name is Grace, and today I'll be talking about a metabolomics approach to increasing CHO cell productivity. This is a quick overview of what I'll be going through in my talk. I'll start with a little bit of background, then I'll take you through some of the major steps in using this metabolomics approach, from an untargeted experiment to identify some metabolites of interest, then to confirming those metabolites in a follow-up targeted experiment, as well as an add-back study. Biologics, usually produced in CHO cells, are a major category of therapeutic drugs, accounting for 7 out of the top 10 drugs by revenue last year, with this proportion poised to increase as the investments made from in the past few decades come onto the market. Advanced next-generation technologies enabling more efficient production can allow even greater access. With this increased use and growing competition, there is constantly more pressure for faster, cheaper production. Now, some CHO cell processes have been reported in the literature to reach impressive titers upwards of 10 grams per liter. These processes have greatly benefited from a lot of work, resulting in increased cell densities and sustained viability. So throughout the process, they've grown and maintained a greater number of cells, making protein. However, even with technologies such as perfusion, there will be a ceiling of cell density. To continue increasing titer, the other lever that can be manipulated is cell-specific productivity, or QP. As opposed to cell growth and viability, QP has had more limited improvement and is not as well understood. We saw a need for a systematic investigation into how QP could be increased in CHO cells. The way we chose to approach that problem was through a metabolomics strategy. Metabolomics has been gaining popularity in analyzing CHO cell processes, but most of the applications have been in investigative troubleshooting. So after a batch does not perform as expected, whether it's lower titer or doesn't meet the quality specs, you might do a metabolomics study on your routine samples to find clues as to why that batch had irregular performance. It's also been used in descriptive characterization, such as to investigate what pathways are generally important in CHO cells. But there are only a very limited number of examples where it has been used to find rational interventions in a proactive way. We've seen very recently, for example, some genetic modifications to CHO cells in order to improve the process, but those modifications were largely to improve BCD and viability. We've not previously seen any demonstrated interventions specifically targeting QP. So in this work, I want to show you how we used a metabolomics approach to come up with 2 practical ways to increase QP that can easily be implemented in process development. First, at the clone selection stage, and second, in media optimization. With this approach, the first thing that we did was gather a library of 12 diverse clonal cell lines. These were all generated through random integration from the same parental GS knockout to produce either monoclonal antibody A or MAB-B. As you can see, when they're run through an identical platform production process, we get a wide range of growth and productivity phenotypes. The peak VCD ranges from about 18 to 40 million cells per mil, and there is a wide range of QPs among the 12 clones. We calculated QP starting from around day 7 or 8 to day 11 or 12, whichever happened to be more convenient for sample processing and analytics. This time period is usually when the cells are most productive. For our metabolomics, we took supernatant samples at a mid-exponential phase and an early stationary phase, with the goal being to identify extracellular metabolites that are present earlier on in the process. This minimizes sample noise caused by cell apoptosis towards the end of a production process, and potentially gives us more of an opportunity to act before the end of an entire 14-day run. An untargeted LC-MS-MS experiment was performed on these samples. This was done on a QTOF with both a hydrophobic column and a hydrophilic column, and in both positive and negative modes for a total of 4 methods per sample for a greater coverage of metabolites. At this point, we had 3,000 to 4,000 features, and of course, feature annotation is a major hurdle in metabolomics. Here we used a tool developed in the Lee Lab at Tufts by my colleague Nick called BioCan, short for Biologically Consistent Annotation. BioCan basically aggregates annotation from multiple databases, such as Metlin or HMDB, and lets them vote so you end up with a consensus score. Then it matches those results with the genomic network of your organism. So, for example, all of the annotated metabolites in this study can be produced by CHO cells. Using this tool, we were able to putatively identify 167 metabolites, although about half of them were duplicates from the multiple LCMS methods. Here we have the area under the curve for each metabolite, normalized to VCD for the day 7 samples from each clone, going from lowest to highest in QP. Day 4 samples did not show any significant patterns, so we focused the rest of the work on day 7 samples, and you can see here that there are some correlations. In order to quantify that, we calculated Pearson linear correlation coefficients between the metabolite levels in QP and between the metabolites in growth, and those correlations are shown on the right. In blue, we have metabolites significantly correlated with only growth with a p-value of less than 0.05, and these were all negative correlations. In orange are metabolites correlated with only QP, and these were all positive correlations. Finally, in green are metabolites correlated with both. We're mainly interested in the group correlated with only QP, as ideally we would like to be able to enhance QP without affecting growth, but to cover our bases, we still considered metabolites that were associated with both when performing pathway analysis. So at this point, we have lists of metabolites significantly correlated with QP, growth, or both. The first practical application we wanted to investigate is whether any of these metabolites could be used as easy, quick indicators of QP. So you can measure the amount of that metabolite or panel of metabolites on day 7 and have a good idea of whether that clone is going to have a high QP or not by the end of a run. And this could speed up your clone selection process. Furthermore, you could possibly identify clones that have the potential for high QP but might not end up with the highest titer because of suboptimal screening conditions, such as running out of nutrients towards the end of production. To test this, we selected a new set of clones to produce a different monoclonal antibody, MAPC, and ran them in the same production process, collecting the day 7 supernames. We got as many chemical standards as we could from a list of significantly correlated metabolites and set up a targeted mass spec assay on a triple quad, which we performed on these new samples. Then we did the correlation analysis and got 2 positive hits. You can see that the correlations hold in this validation set below, showing that aspartate and cysteine concentrations are predictive of QP in a completely new set of clones. Of course, this is only 24 clones, so further testing would be warranted to have more confidence, but here's a proof of concept that you could use this approach to find a biomarker panel for QP. Now we come to the second practical application, finding metabolites that could potentially be correlated with QP because their presence is directly beneficial for the cells, and adding more would be even better. We took the list of all metabolites correlated with QP and did a pathway enrichment analysis using MetaboAnalyst. I want to mention, since these are extracellular samples, you might wonder, are these increased levels in the supername from accumulation of media components, or are they actually products of cell metabolism? So we went back and checked and found that a majority, 75% of the metabolites from our list, were not in the chemically defined feed or basal media. So they are actually being produced by the cells. Now, going back to the pathway analysis, we had several pathways come up as significantly enriched in our list of metabolites. So above this dotted line indicates a p-value of less than 0.05, and 2 particular pathways that were both significantly enriched and saw a high pathway impact were the TCA cycle and alanine, aspartate, and glutamate metabolism. From here, we chose some metabolites out of these 2 pathways as our candidate metabolites for an add-back screening study. Those metabolites were citrate, aspartate, glutamate, and GABA. We performed a 96-condition DOE study in 50 ml conical tubes with the highest and lowest QP clones for each of the initial molecules. 3 levels of addition for each metabolite were chosen, with the lowest level based off of either the maximum amount measured in that untargeted experiment or the amount that is already present in our platform media if it's there. One compound did turn out to have a direct effect on the culture performance. So looking at all the conditions colored by citrate concentration, there appears to be a trend. While if you look at the conditions colored by any of the other metabolites, for example, aspartate, there is no clear trend. So we wanted to examine the citrate effect a little more closely. This is the same data, but separated by clones and pulling out peak VCD to represent growth and looking at QP calculated across days 7 through 12. For growth, we see first that clearly the high level here in red was too high. It negatively affected the peak VCD. For some of the clones, even the low level had a detrimental effect, but there were also many conditions where the peak VCD was within 10% of the control. Looking at QP, addition of citrate did improve QP for several clones, but what we really want is QP improvement without lowering the VCD, and we did see that with this second clone. So a higher QP without impacting the VCD gets us to that higher final titer. We wanted to follow up on this in actual bioreactors with DO and pH control, so we did a confirmation experiment in an amber 250 using a narrower range of concentrations, since this highest one was clearly not helpful. We used 6 clones this time, and were able to replicate the dose-dependent improvement in QP with citrate addition, which we saw in 5 of the 6 clones. For one of these clones, clone B1, there was also that impact on VCD that negated the improvement when it came to overall volumetric titer, but for the other 4, we saw a 15 to 107% increase in titer at the highest level, including clone A3, the one that started out with the highest titer. So I want to point out that this study illustrates the possibility of rejecting a good clone because of suboptimal media conditions. Now, of course, we can't optimize everything at the clone screening stage, but if you have learned from an approach like this that many clones generated from your parental cell line do well with a certain additive, you may want to include that in your screening design. Now, we don't know exactly why adding citrate increases QP, so that would be an interesting avenue for future study. Potential explanations could be that it provides a chelating effect, or it simply provides additional flux through the TCA cycle. Similarly, with the aspartate and cysteine QP indicators, we're not sure why these are correlated with QP, so it would be interesting to use other omics tools to look into potential mechanisms. Additionally, we have seen these significant correlations across 2 sets of 12 clones, but if we're able to repeat this with more clones or even expand to other CHO cell lines, we would really be able to confirm these metabolites as biomarkers of QP. So to recap this metabolomics approach, we first selected a diverse library of CHO clones with a wide range of QP. They are all from the same parental cell line, so this approach depends on your initial library, and if you want to find metabolites across multiple CHO cell types, you could start with them all in your library, or you could use whatever is on hand, but then later do confirmation testing in other cell types. Next, we did an untargeted metabolomics approach in correlation analysis to find metabolites significantly associated with QP. And finally, we performed follow-up experiments with those metabolites of interest. This led us to find some compounds which were correlated in a separate set of clones that had potential use in clone selection for early identification of high titer clones, and that other compounds could be used directly as media additives to improve QP and titer. So ultimately, we were able to successfully use a metabolomics approach to develop rational interventions to improve QP. I'd like to acknowledge my colleagues at BMS, Katie Arian and Mike Boris, and Professor Lee from Tufts for their support and guidance, as well as Nick, who developed the annotation tool I used, and many more colleagues who provided help along the way. Thanks to all of you for your attention.